So, talking about different chemolithotropes, let's start with the sulfur oxidizing bacteria. These sulfur oxidizing bacteria use sulfur as the sulfur containing compounds as the main oxidizing compounds, which give rise to which acts as the electron donors. They leads to the oxidation of, for example, sulfur, oxidation of hydrogen sulfide, oxidation of thiosulfate. All of these produce sulfuric acid, and because of this production of sulfuric acid, this really has some ecological importance, some environmental importance also. There are two modes of ATP production in these microbes. One is the substrate level phosphorylation. You have already studied and know about the substrate level phosphorylation. You will also discuss and study the substrate level phosphorylation when you will be studying, or you have already studied, the glycolysis process, in which substrate level phosphorylation gives rise to the formation of ATP. Similarly, substrate level phosphorylation gives rise to the formation of ATP here also in these sulfur oxidizing bacteria. The key molecule involved is the APS, adenosine 5-phosphosulfate, not phosphate, sulfate. They form from sulfide and adenine monophosphate. So sulfite is the major component here. It's a high energy molecule. That's why it is used in the energy production. Then there is the oxidative phosphorylation. Again, you must be familiar with this term. The oxidative phosphorylation is the type of phosphorylation in which oxidation occurs, and it takes place in mitochondria. It's a very in eukaryotes, in higher eukaryotes, in us for instance, in mammals for instance, this is a very successful energy producing or very efficient energy producing mechanism. And as far as the sulfur oxidizing bacteria are concerned, here they use the sulfur containing compounds like thiosulfate, like hydrogen sulfide. These compounds are used as the major electron donors, which then takes place, which then carry out the electron transport chain, and which give rise to the energy eventually. One thing you should keep in mind, that one of the major principles of electron transport chain is that electrons have to be donated between two moieties, between two components. If electrons move from component A to component B, there should be a very high electronegativity difference between these two, between the two molecules. But here the sulfur doesn't have that much that much difference, or sulfur containing compounds don't have that much difference. They are not as much negative to give rise to the electrons, which can be easily be donated and which can easily be carried through the electron transport chain. So these molecules, these microorganisms have devised a way that electron transport chain can somehow be carried out, and the way through which it is carried out is to use the energy from the proton motive force, the energy you get from the transportation of the hydrogen ion. That energy is utilized in the transportation of electrons from the donor molecules like sulfide molecules or sulfur containing compounds. So because this occurs not from a very highly negative moiety towards the highly positive or relatively positive, it occurs from a less negative moiety. So we say it's the reverse flow of the electron. So in these microorganisms in which oxidation of sulfur, and you will see also in case of oxidation of iron also, that the reversal of the electrons are transferred in a reverse direction, and this is made possible through the proton motive force. Also, sulfur oxidizing are very, very flexible. They can also they also can grow in the aerobic environment, and they can also grow in anaerobic environment. When they are in an anaerobic environment, then of course the oxygen is not present. Then the sulfur component acts as the major electron acceptor. In generally when we are talking about aerobic respiration, as it's occurring in us, the oxygen is the major electron acceptor. But here, in case when they undergo anaerobic respiration, these microorganisms, they use other moieties to act as the major, the electron acceptor, and here the major electron acceptor is the sulfur. They can use, as I've already told you, that they can also act as autotrophs, so they can use carbon dioxide as the major carbon source, but for that they have to depend upon Calvin cycle, and which again is very energy consuming process. And the main carbon source, if they are not autotrophs, they are heterotrophs, then they will use the organic moieties like the glucose or like amino acid and so on and so forth. Then as I have already told you, that because they produce the sulfuric acid, and because they are responsible in maintaining the sulfur content in your ecosystem, so they have some real environmental importance, and they which also show that how they are modified, or they are made adaptable to different environments. They show the diversity in their adaptability. They can be aerobic, they can be anaerobic, they can be autotroph, they can be heterotroph, and this shows that how flexible they are in their metabolism. And it also shows how successful microorganisms can be. That's why if you go to any part of the earth, no matter how extreme are the conditions there, no matter how hostile are conditions there, you will see the microorganisms still thriving there. And this is because of the flexibility in their adaptability, which we are discussing.